Welcome everyone. Good afternoon or good morning, depending on where you are on the globe. My name is Thomas Hureau. I'm from the University of Strasbourg in France, and I'm glad today to host this virtual journal club from the Journal of Physiology and the Physical Society. And today we are going to talk about a paper entitled Sex Differences in Fatigability Following Exercise Normalized to the Power-Duration Relationship. And today I'm happy to be with authors Kirsty, Hicks and Paul Ansdell from Northumbria University. They will be here as panelists and they will present their interesting paper. So before to start, on behalf of the Journal of Physiology, I have few information. If you want to get involved with the journal, you can host a future meeting of the Journal of Physiology's virtual journal club, what I'm doing today. You can also write a journal club article on a recently published article in the Journal of Physiology. Also, you can submit your research to the journal. The Journal of Physiology publish original research papers in all areas of physiology and pathophysiology. So if you have any question, you can email the Journal of Physiology or the Physiological Society. So before to start now, let's look at the session outline. First, I will give an introduction on this paper and topic. Then the authors will present the selected paper. So Kirsty and Paul, thank you for accepting my invitation for this event. Then we will get some feedback from the editors. So unfortunately, the editors are not available today, but I got their feedback to give and to try to tell the audience why this paper was selected for publication in the Journal of Physiology. And then the last part of this journal club will be a Q&A session, question and answers for about 10 minutes. And this will be followed by a networking session. So please, if you are interested, click on the link into the chat at the end of the journal club. So a few more information before to start. You can submit your question at any time during the presentation using the Q&A box. So don't wait for the end of the presentation and please ask at any time. Also, you can please add your name and institution if you want to, but if you prefer, there is also a tick box to submit anonymously. For example, if you want to ask a very tough statistics question. You can also upvote your favorite questions. And please note that the session is recorded and you will get an email within a week with the link to the session associated to a feedback survey. So let's start with the introduction. So this paper will talk about sex differences in general, and I wanted to give a large introduction. So in 2005, it is interesting to note that only 3% of grant proposals measured sex differences. So there is a big gap between what we know about males and females physiology or in general in science. So that's why in 2014, there was a policy of including sex as a basic biological variable for research involving humans, animals, and cells in NIH-funded research. So I thought it was interesting, and you can see that on the illustration on the right of the screen, that this balance between males and females is not only in human research, but also in animals and cells. And why does it matter? It is because, as you know, gonadal hormones have important influences on organs and systems outside of reproduction. So now if we move on the sex differences in exercise-induced fatigue, the topic for this paper today, you can see the numbers and ratio of men and women in 200 fatigability studies from 1972 to 2015. And you can see with the black bars that women are underrepresented in these studies compared to men. So the paper of today will try to fill this gap. But another important thing, like a classic question when we try to compare independent groups, such as men and women, but it's also true for young and old population or healthy and some disease population. How are we going to compare them? In absolute or in relative intensity? So the first option is absolute. For example, both groups will exercise at 100 watts. And this is interesting to give some clinical insights, for example, if you're interested in the quality of life or exercise capacity, and especially for daily living activity, such as climbing stairs or doing your groceries, absolute intensity is actually very insightful. Now, if you want also to have maybe more mechanistic insights, the relative comparison is also very interesting. But the question now is relative to what? And there are many options. So very often we can normalize to the percent of MVC, maximal voluntary contraction, percent of VO2 max to muscle mass. But in the specific case of comparing men and women, what's difficult is that we have other confounding factors that will independently affect fatigue, such as the differences in maximal force, muscle typology, muscle mass, but also the metabolic rate of exercise. So that's the idea of today. In the Journal Club article for today, exercise intensity was normalized to critical power, so to a metabolic threshold. And you can see on the screen on the right, the level of peripheral fatigue evoked by supramaximal electrical stimulation over the femoral nerve. It is actually fatigability because it's in Newton meters per minute. And you can see on the x-axis the exercise intensity. So it is torque, percent of maximal voluntary contraction. And so exercise was done below or above critical power or critical torque, depending on the type of exercise. And you can see very different kinetics. When you exercise above critical power, which delineates the severe domain of exercise intensity, you have a linear relationship between the fatigability and the intensity of exercise. However, when you go below critical power, which is now the heavy domain of exercise intensity, you can see that fatigability is not on this line anymore. It is very, very small. It's not nothing because they exercise for a very long time, so they still develop some fatigue, but definitely the rate is very, very different. And it is relevant because, as you know, peripheral fatigue is linked to the intramuscular metabolic milieu of your locomotor muscles. So the idea of the paper from Kirsty and Paul will be to normalize exercise to this metabolic threshold. So I'm really looking forward for their nice presentation. And so Kirsty and Paul, please go ahead.
Thank you very much, Tom, for the introduction, and thank you for the invite to present our paper. So I'm going to share my slides now, so you guys should see that there. Okay, so yeah, this presentation for the next 20 to 25 minutes, we're going to talk about the rationale for our study, the methods we employed, and the results and their implications. So to start with, it's important to note that the paper we're going to discuss today was an extension of previous work. So previously we had studied sex differences in fatigability relative to the intensity duration relationship, and this was during single limb exercise. So as Tom just stated, critical torque is the metabolic threshold that can be assessed in that exercise modality. And we showed that females had a greater relative critical torque, so when expressed as a percentage of their maximum voluntary contraction compared to males. And then also when exercise was normalized to that threshold, females fatigued at a slower rate. So that was single limb exercise. That's not why we're here in this webinar. We're going to talk about whole body exercise. So the paper we're discussing today used cycling as the exercise modality. And the power duration relationship and critical power during cycling is determined by, put simply, the oxygen transport pathway. So whether that is convective capacity, so ventilation within the lungs, the diffusion of oxygen into the bloodstream, cardiac output, or the diffusion of oxygen out of the blood into the working tissues. So it's we term that collectively as convective capacity, but critical power is also mediated by the diffusive capacity of the muscle as well, and whether that's capillarization, mitochondrial density and function, and muscle fiber typology as well. So if there's any sex difference at any stage of this oxygen transport pathway, there's the potential for the power duration relationship to be different when expressed relatively. So if we take a look at the individual physiological systems that contribute to the oxygen transport pathway, let's start with skeletal muscle. It's fairly well accepted that females typically demonstrate a greater proportional area of type 1 muscle fibers for an equivalent training status. And with that, we expect things such as greater capillarization. It's been demonstrated that females have a 30% greater mitochondrial respiratory capacity, and also a greater fatigue resistance within the skeletal muscle. So this potentially benefits in the diffusive capacity of the oxygen transport pathway there. When we look at the respiratory system, on the other hand, males typically demonstrate larger lung sizes and airway sizes, even when matched for height compared to females. And due to these differences in lung size and geometry, males have a lower mechanical work of breathing and a lower oxygen cost of breathing at high minute ventilations. When you combine this with the lower concentrations of hemoglobin, typically found in females compared to males, it puts women at a greater risk of exercise-induced arterial hypoxemia. And put simply, if we balance up the convective and the diffusive capacity of males and females, it potentially looks something like this. So females might have a greater ability to utilize oxygen within the working muscles. However, there may be a lesser ability to transport oxygen. So you can begin to see how the oxygen transport pathway, and therefore the metabolic thresholds during exercise, may be mediated by sex. So I'm going to pass over to Kirsty now, as she's going to talk us through the study design.
Okay, so based on the information that Paul's just taken us through, the study had two main primary aims. The first primary aim was to compare the power duration relationship between males and females during cycling. To answer this question, 10 males and 8 females were recruited, and it's keen to note that the female participants had been on hormonal contraception for at least the 6 months previous. Participants attended the lab 6 to 7 times. Initially, participants were required to perform an incremental exercise test, and this was to calculate their VO2 max and their maximum power, referred to as P max. This test was conducted for 2 reasons. Firstly, to confirm the inclusion criteria of either a VO2 max above 55 or 48 for males and females respectively, or a P max of 4.6 and 3.8 watts per kilogram for males and females respectively. Secondly, this initial test was carried out to obtain P max, which was used to then calculate the intensities required for the constant load tests. So following the ramp test, we needed to estimate the parameters of the power duration relationship, critical power and W prime. To do this, we got the participants to perform 3 constant load tests at 80, 90 and 110% of their P max in a randomized order. Throughout this test, pulmonary gas exchange was recorded continuously, and this was to ensure that the VO2 was within 95% of their VO2 max. All participants achieved this, and all time to task failure occurred within 2 to 15 minutes. Very briefly, the critical power and W prime were calculated using 3 equations: the inverse linear model, the linear work time model, and the hyperbolic model, with the highest R squared and the lowest standard error being the dictating which equation was taken forward. As Paul previously mentioned, due to the trade-off between poor convective and greater diffusive capacities in females, our hypothesis to this aim was that there would be no difference in the power duration relationship when expressed relative to maximum exercise performance. So if I now go to study the aim 2 of the study, our second aim was to determine whether a sex difference in fatigability, represented as time to task failure, and the integrative mechanisms such as neuromuscular, muscle oxygenation, and pulmonary gas exchange existed when exercise intensity was normalized to the power duration relationship. The way in which we set out to explore this was following the calculation of the power duration parameters, which I've mentioned previously, critical power and W prime. Participants completed a constant load time to task failure, which was performed in a randomized order at 110 or 90% of their critical power. This placed our participants in either the severe or the heavy intensity domains. For the severe domains, participants were just asked to exercise until failure, whereas within the heavy, participants were asked to exercise until failure or for 60 minutes, whichever occurred first. It was hypothesized that when exercise was metabolically matched, time to task failure would not differ, but females would exhibit greater fatigue resistance and less deoxygenation in the knee extensors. Okay, so I keep mentioning this integrative physiology. But how did we actually go about measuring that in the lab? And I think this picture here on the slide really gives us a nice overview of our testing protocol, because we used multiple methods and protocols to measure the convective and diffusive contributions to whole body exercise. Prior to the exercise, neuromuscular function, which Paul will go through in detail in a moment, NIRS, MVC were all measured at rest. Breath by breath pulmonary gas exchange was measured throughout the throughout the time to task failure tests. More specifically, minute ventilation, oxygen consumption, carbon dioxide production, and RVR. Additionally, throughout the protocol, NIRS was measured on the vastus lateralis to measure oxygenated and deoxygenated hemoglobin, and also tissue oxygenated index, the TOI. NIRS was analysed retrospectively at 20, 50, 75, and within the final 30 seconds of the time to task failure. I'm going to pass to Paul now, who will just talk you through the neuromuscular measures.
So yeah, as Kirsty mentioned, pre and immediately post-exercise, we quantified neuromuscular function and fatigability with non-invasive neurostimulation. So we began with maximum voluntary contractions superimposed with supramaximal electrical stimulation of the femoral nerve. This gave us our index of voluntary activation and the potentiated twitch. We then assessed corticospinal excitability using transcranial magnetic stimulation and subpercutaneous electrical stimulation of the lumbar regions of the spinal cord to allow us to quantify cortical and subcortical contributions towards corticospinal tract excitability. These stimulations were performed at 10% of maximum voluntary contraction. We also assessed paired pulse TMS responses in the form of short interval cortical inhibition, or SICI.
Okay, so what did we find? Well, if we think about this in relation to our first aim, during the power duration relationship, we can see here that the absolute critical power was significantly higher in males compared to the females. And we can see that here with 260 versus 179 watts. However, what's really interesting is that when we make this relative, we can see that that sex difference completely null and void, and there was no significant difference, 72 and 74% of the maximum power. If we now take a look at fatigability, and we're going to focus first of all within the severe domain, okay? There was no significant difference in the time to task failure, so there's no difference in the exercise length. Despite this, females exhibited a lesser decrease in potentiated muscle twitch, with a difference as high as 50% compared to their male counterpart, which you can just see here with 36 versus a 15% decrement. There was no other differences in any other neuromuscular fatigue measure. If we look in the heavy domain, we can see similar fatigue findings, with women again demonstrating a lesser decrease in the potentiated muscle twitch, demonstrated in the figure on the right-hand side, but there was still no other significant differences in any of the neuromuscular fatigue measures.
So if you remember back, we took a few other measures during cycling in these two intensity domains. And if we look at the NIRS findings here, and this is tissue oxygenation index expressed as a percentage of resting baseline, we can see that females in the red had a lesser decrease in muscle deoxygenation during both severe and heavy intensity exercise. If we move on now to our measures of corticospinal excitability, one thing that was interesting, or I found it interesting at least, was that corticospinal excitability decreased in the heavy domain only. So on the left-hand side here, we have the MEPS and LEPS, which are lumbar evoked potentials. Neither of these two parameters decreased pre to post exercise in the severe intensity domain. However, both parameters decreased by about 20% compared to baseline following heavy intensity activity. And there was no statistical sex difference in this decrease. So to sum up this study, our conclusions were that when oxygen delivery becomes a limiting factor as well as oxygen utilization, there's no sex difference in the maximal sustainable work rate. And it's important to know that this is when expressed relative to the individual's maximum capacity. So perhaps the superior oxygen utilization is counteracting lesser oxygen delivery in the females. When exercise intensity was matched, and if you remember, we did this 10% above and below critical power, females experienced less muscle fatigue. And additionally, female quadriceps deoxygenated less in both the severe and the heavy intensity domains. So we have a few thoughts on this data, and as my supervisors liked to remind me during my PhD, so what? What does this mean? The first kind of thought that popped into our head after looking at these data was, why didn't females exercise for longer if they had less muscle fatigue? And it's a difficult one to put our finger on, really. But I go back to this quote from David Poole's work, that the nature of W prime cannot be limited to a single physiological process. So we typically think exercise tolerance in the severe domain is limited by neuromuscular function or peripheral fatigue, however we want to term it, or reaching a critical threshold of metabolic stress within the working muscle. It's possible that cardiopulmonary stress also contributes to W prime. And as we saw earlier, there's the potential exists that the amount of limitation, stress, and therefore afferent feedback from different physiological systems could contribute to W prime and exercise tolerance differently in males and females. So to put this into context, it's been demonstrated. I think there's work from Bill Shields' laboratory talking about how the perception of dyspnea differs between males and females, and it's a more severe perception for a given ventilatory rate in females. So what we think might be happening here is that all of these physiological or afferent feedbacks from different physiological systems contribute to the maximum tolerable symptom of fatigue, or as Thomas has put it previously, the sensory tolerance limit. And what we think might be different between males and females is the magnitude of the different contributions from different physiological systems might be different in males and females, whereby the tolerable symptom of fatigue is reached predominantly from afferent feedback from the muscles and the peripheral fatigue generated in males, whereas in females, there's less afferent feedback from the muscle and greater afferent feedback from the more central systems.
I think what I really like about this study is that we answered several questions, but we raised more questions than we answered, and especially in the acute implication. So if we just recap on our kind of salient finding that there was no sex difference in the times task failure in either intensity domain, yet females exhibited a lesser reduction in knee extension function immediately after exercise. So to rephrase that, for an equivalent dose of exercise, females experienced 50% of the muscle fatigue experienced by their male counterparts. Now, this finding remains true for both our single limb and the current whole body study. But what does this mean for our athletes? We know we need a metabolic disruption to activate the cascade of pathways required to achieve adaptation. And Paul is going to go through this in the next slide in a moment. But before we go on to these stressors, the question we are asking here is, are we stressing the female athlete enough? If we're using the equivalent rest, intensity, and volume configurations between male and females, are we maximizing the potential of our female athletes? Or are we training them in a constant state of understimulation? We don't know, is the simple answer.
So yeah, as Kirsty mentioned, unfortunately for us, we do need fatigue and the unpleasantness that comes along with fatigue in order to adapt in the long term. So we know that metabolic stressors such as hydrogen ion accumulation, cellular hypoxia, ADP and AMP accumulation, and reactive oxygen species are all stimuli for signaling pathways that lead to mitochondrial biogenesis and angiogenesis, and all the great adaptations that endurance athletes crave. So the question exists now that we'd like to answer in the future: do females receive half the adaptive stimulus for the same dosage of exercise? If we're making the link between muscle fatigue and adaptive stimulus, this is. So there is some preliminary evidence here, and I'd point you in the direction of this Scalzo paper from 2014. So the authors employed 4 weeks of sprint interval training in males and females, and they measured muscle protein synthesis and mitochondrial genesis pre and post intervention. And interestingly, what they saw was in both parameters, whether it was muscle protein synthesis in the cytosol or the mitochondria, there was a substantially lower increase in females compared to males, And again, mitochondrial biogenesis was significantly lower in females compared to males. One study is definitely not enough for us to base our conclusions on, so there's definitely scope for further research in this topic and area, and it's hopefully something we'd like to be able to answer in the future. And obviously we are limited for time, so if you would like to read more about our thoughts on this matter and the acute and chronic implications of physiological sex differences, I'd point you in the direction of this review paper that we were fortunate enough to write last year. So just to wrap up our presentation, I'd like to say thank you for listening. Thank you to my PhD supervisors. This paper was the culmination of 3 years of PhD work for me. Thank you to our collaborators, Professor Sandra Hunter, Jakob Škarabot, and the master students who helped me out with data collection, because without these guys, you wouldn't have seen half the data that we presented today. So thank you very much. I'm going to stop sharing slides there. Happy to answer any questions.
Okay, thank you very much, Kirsty and Paul, for this really interesting presentation about your paper, but also about your future direction, which is always nice to hear about. Mm-hmm. We are now entering to the third part of this journal club. So I will give some feedback about the editors' comments about what made this article selected for publication in the Journal of Physiology from an editor perspective. So unfortunately, both editors, Professors Bruno Grassi and Michael Hogan, were not available today, but they gave me their feedback on the paper. So as you may know, there are 2 layers of editors in the Journal of Physiology, a senior editor and a reviewing editor. So that's why they both gave me some feedback. So actually, there are like three key points about why this paper was really interesting for the journal. First is the topic. Sex differences is still under-investigated in physiology and in science in general, so it was interesting for the journal to also push that kind of question. The second key point is the novelty of the study, with the normalization of exercise intensity to critical power during whole body exercise, which extends previous findings from single joint isometric contraction, also published in the Journal of Physiology by the same research group. So that's the second point. And the third one is the comprehensive physiology approach of the research question that led to important mechanistic insights, as you saw with our presentation, with different methods and different techniques. So taken together, it was recognized by reviewers and editors that this investigation had the potential to make a major contribution that advances the field. So for this reason, this paper was worth it to publish in the journal. So now we are entering in the last part of the journal club with the Q&A session. So please use the box to put some questions, and you can also again upvote your questions. So I'm sure Kirsty and Paul will be happy to answer everything.
Absolutely.
Maybe I will start with a first question. So a little bit about it, but what I think is very interesting is that there is a difference in fatigability between men and women. However, exercise time to task failure was similar. So my question would be, why don't the women go further? Why don't they reach the same level of fatigue than men? So it's more like about probably exercise limitation and task failure.
May I take that one, Kirsty? Cool. So yeah, I think it goes back to if we think about exercise tolerance and fatigability and how it limits performance, the sensory tolerance limit and variations thereof. So, you know, the sensory tolerance limit, or the maximal tolerable symptom of fatigue, can be a result of multiple contributing systems and feedback from those systems. Therefore, what I think's happening, or what we think's happening in this situation, is that at task failure, there's a different contribution from different physiological systems in males and females. So if we think about the exaggerated muscle fatigue in the males, this is what we typically think limits severe intensity exercise performance, and we reach that kind of critical threshold of peripheral fatigue or muscle dysfunction. And what we think is happening in this situation is that there's less contractile dysfunction due to differences in muscle fiber typology between males and females and the other physiological differences. However, there may be greater kind of central limitations, and the feedback from those central systems in females that also contributes to the maximal tolerable symptom of fatigue.
Okay, thank you very much. So we have a first question from the audience. And again, you can keep going with asking your questions. This is a question from Catherine Notarius from the University of Toronto. So any thought on how this would impact clinical populations limited by both convective and diffusive factors, such as heart failure?
Don't mind taking them. Cool. Yeah, I guess the results of our findings, it's yet to be seen whether they translate into clinical populations. We had healthy, trained males and females. I think if the sex differences in both those systems still exist, there's a rationale for differences in fatigability. I guess the question is whether things such as chronic heart failure or other disease states are a bigger stimulus than the sex difference itself, and whether chronic heart failure causes so much of a disruption in convective capacity, and I guess later down the line in diffusive capacity as well, whether the heart failure stimulus outweighs the sex difference.
Yeah, I think I'd add the same, is that because we can't account for all contributing confounding variables in that population, it might be that those small or medium magnitude differences we're seeing with the sexes, they may be nullified by other issues, shall we say, that the clinical population face. I mean, it's a fantastic and really interesting question, but I think we've got to find a way to really home in on the mechanisms to be able to answer that kind of clearly, I think.
Mm. I think just one more thing is chronic heart failure is characterized by oversensitization of afferents, muscle afferents as well. So perhaps if females are experiencing less muscle fatigue, there may be different magnitudes of afferent feedback. And if we're going back to the sensory tolerance limit, there could be an interaction between afferent feedback and sex there, potentially. I don't know the exact answer, I'm afraid.
So the next question is from Rafael Azevedo. So there is not the affiliation, but if I recall it, it's from Brazil. Considering the lower mitochondrial adaptation in females, is it because they adapt less or they already have a great mitochondrial capacity compared to males?
Yeah, you can take it, Paul.
Yeah, I think, yeah, it could be a result. So there's the paper by Corden A, I think, I don't know if I'm pronouncing that name wrong, I apologize, but showing that females of equivalent training status have greater mitochondrial respiratory capacity. So it could just be a result of that, and that there's more of a ceiling effect perhaps in female mitochondria, or it could be that for the reasons we stated, there's less of an adaptive stimulus. It'd be really interesting to pick that question apart experimentally, I think.
Okay, so the next question is, how do you think that the menstrual cycle can influence fatigue mechanics and fatigue tolerance?
And if I take that, Paul?
Of course.
Yeah, I think, again, it's a really interesting question, and it's probably one that our lab group have discussed at length over the last, well, probably 3 years, especially the last 18 months. And if we go back to Paul's first slide where he discussed the opportunity from the kind of convective through to the diffusive capacities, there are sex differences at all changes of those that may influence the whole integrative response, either in single limb or to whole body. And the same theory can be applied to the menstrual cycle. There's estrogenic and progesterogenic effects at each possible pathway along that. So theoretically, there is a possibility that the menstrual cycle does have an effect. However, as we know with the recent reviews by McNulty and Elliott Sale, there's trivial effect across the menstrual cycle at the best of time as performance in its global capacity. So although it seems a bit of a cop-out answer, at the moment, we're not entirely sure what the menstrual cycle may do to these. It would be a very, very interesting study and one that we would love to answer in the not so far future.
Yeah, and just on top of that, the first paper from my PhD was a single limb fatigue paper where we saw that in an open-ended exercise task, we normalized to 60% of MVC, that females could exercise for longer in the luteal phase. And I think we weren't able to work out whether that was a result of critical power changing or W prime changing. However, again, I think that would be a really interesting study to run and see if the power duration relationship is affected by menstrual cycle.
Okay, great. So were the blood pressure and heart rate responses at both intensity also lower in female participants at both intensities?
Yeah, unfortunately, we tried to record beat by beat blood pressures with the Phenopress in this study, and we successfully did it in our single limb study that we mentioned at the start. However, we weren't able to accurately collect data or use this technique in the cycling modality. So we haven't got that data, unfortunately, for this paper.
Right. So we have a question from Simone Porcelli, who first congratulate you for the nice presentation. And he wants to ask, do you have any insights from your data about potential differences for intermittent exercises in fatigability between male and female?
Fancy this one, Kirsty?
I think for me, it's a really interesting question, but it does vary differently between the modality of exercise. So it would be one that, although we can infer findings, we wouldn't be able to kind of produce conclusive findings from this study. I think the intermittent exercise is something that we would like to go further down the line of, especially manipulating those configurations of rest and recovery between the sexes. But again, it's one of those that's up and coming, I think, and it is really interesting. I don't know if Paul wants to go into the differences in the continuous versus intermittent fatigue response we looked at as well.
Yeah, I think if we're referring to intermittent exercise as, let's say, like football performance and intermittence that way, you know, you can apply the power duration relationship to an intermittent model. So perhaps if W prime is used up differently during intermittent exercise, or if females can recover W prime quicker, then there's potential for there to be sex differences in the fatigue response in that modality as well.
Definitely, and I think it's something that I've seen in a practitioner-wise as well, the same kind of stimulus prescribed, whether using a maximum aerobic threshold or whether using a critical torque threshold, we do see anecdotally differences in the response in males and females, despite trying to make that session stimulus metabolically matched. So anecdotally, I'm seeing the differences. Experimentally, we've yet to explore that.
Okay, so the next question is from Hugo Kerhervé from the University of Rennes in France. Did you observe faster kinetics in NIRS for female, as in Muria's group NIRS papers, during the square wave exercise?
So we didn't look at kinetics, onset kinetics in this paper. I think if we wanted to answer that question, we'd need to set the study up a bit different experimentally. So perhaps have multiple square waves of exercise in order to look at the onset kinetics. It would be a really, really interesting question to ask. It's something we wanted to do last summer. Unfortunately, for obvious reasons, we weren't able to get in the lab. But again, you might hypothesize that at the peripheral level, so within the NIRS signal, you might see faster onset kinetics due to the differences in that diffusive capacity between males and females.
Okay, so we have now a question from Gregory Perse, asking, would you expect that blocking peripheral afferent feedback of the exercising limbs may shed some insights on the sex differences? For example, a greater benefit for males than females.
Yeah, definitely. And
Northwestern University.
Yeah, definitely. And some of the kind of afferent blockade studies are of particular interest here. And if we're talking about the sensory tolerance limit and the different contributing factors, it would be interesting to perhaps, I don't know if it's experimentally possible, but blockade the afferent feedback of individual physiological systems. So if we blockaded the feedback from the respiratory system, for example, does this allow females to exert their muscles or experience more contractile dysfunction before task failure? That would be a really interesting way to kind of explore this and our speculations in a much more detail, yeah.
I think ethics might be a hard hoop to jump there.
Maybe once we're allowed back in the lab, who knows?
Yeah.
Great, thank you. So Massimo Venturelli from the University of Verona is asking, do you think that in females there is a potential limitation in the blood flow to the exercise limb?
Yeah, it's interesting. It wasn't something we were able to explore with our data set in terms of limb blood flow. But I know there's some interesting data looking at things like the metaboreflex when the lungs are stressed, and it appears that females have a lower metaboreflex and lower limb cardiac output is greater preserved in females compared to males. For the life of me, I can't remember the author's name, but there's a paper showing a greater femoral artery dilation at the onset of exercise in females compared to males. So perhaps at the peripheral level, blood flow isn't a limitation. It might be that the central limitations perhaps and the lesser hemoglobin concentrations that contribute to the central limitations in females. I hope that answers your question.
Okay, thank you. So the next question is from Ren Brocksterman from the University of Utah. So what do you think is causing the lower W prime in females? And do you think this influences how we interpret the development of fatigue during exercise?
Do you mind if I go, Kirsty?
Yeah. I'll just add a bit at the end if you don't cover it.
Yeah, so in absolute terms, males had a greater W prime. And I think the result or the cause of this is because equivalently trained males and females, males will use or produce greater forces and power outputs than females due to differences in size and muscle mass. So in absolute terms, W prime was greater in males. I guess with the similarities in time to task failure, W prime on a relative scale was used up at the same rate, and there was no difference in W prime when expressed relative to P max. One thing, and it came up in the review of the paper, maybe you're the reviewer in secret, I don't know, but one thing that came up was perhaps the sex differences in muscle fatigue was because males simply did more work in terms of joules and worked at higher wattages. So is it simply that males were producing more power and therefore more work over the same time period that caused that sex differences in muscle fatigue? It might be an interesting way to unpick it, to have males and females with similar power outputs and compare those. The thing is, when you compare or if you match males and females in that sense, then you may have a difference in training status of the males and females.
Yeah, because you'd have to have really high trained, well, I assume really high trained women to be able to get on that same equal paring of absolute power. So I think that would be another confounding variable that'd be added into the mix there. But no, I think Paul's answered that fantastically.
Okay, so the next question is from Joshua Carr from Texas Christian University. So regarding potential sex differences in fatigue stress adaptation, would you expect similar sex differences in aerobic versus resistance training? In your opinions, what should the questioning focus?
I think for the resistance training, I think this is what I find really interesting, the potential to adapt in kind of female and male studies. And it's definitely something that needs exploring more, because again, you see differences in rate of adaptation in resistance training and in kind of traditional endurance or HIIT training between male and females. Where the difference is focused, I think there's the it depends on the length of the endurance exercise. So if you start looking into ultra endurance, actually you see if it's equivalent for the amount of participants in ultra endurance that are male and female, you see that females actually can outperform males. I would really be interested to look at how we can optimize strength and resistance training in the females and really maximize those adaptations, because I do think there is significant differences and sex differences in the potential to adapt following strength training. But it requires a really long study and access to labs, so hopefully that's possible. Paul, do you want to add on that regarding aerobic? Or
yeah, I think it's so task dependent, this sex difference in fatigue and the mechanisms of fatigue. So we need to explore whether the mechanisms of fatigue or stress during resistance training are the same in males and females before we could kind of study the long-term adaptability, if that makes sense.
Okay, thank you. So if you don't mind, maybe your last question from Paul Morgan from the University of Birmingham. So your data show that potentiated twitch torque and thus peripheral fatigue is lower in females at the limit of tolerance. Is there any evidence backing this up on phosphorus metabolites, AMP, ADP, H+ accumulation, PCR?
To my knowledge, no, and it's something that would help us explain some of what we're talking about, and definitely help us to make stronger conclusions and speculations. So similarly, if there is a lesser accumulation of hydrogen ions, depletion of phosphocreatine, and all those negative things we associate with the severe intensity domain, whether that underpins the reduction in potentiated twitch, then we could be more confident when we say females may be experiencing less of an adaptive stimulus. Unfortunately, we don't have those facilities in our laboratory, but it'd be really interesting to find out if that's the case.
Okay, thank you. Thank you very much for answering all of that type of questions. So we are now arriving at the end of this journal club. There are still some few questions, but good for joining us for the networking session. So you will get, you just need to click on the chat to follow us, and we'll be very happy to discuss with you more about this paper and probably science in general. So thank you very much for you, Kirsty and Paul, for this very nice presentation. It is always nice to have the authors in the journal club, something that we have generally when we do that in lab meetings. I would like also to say thank you to the Journal of Physiology and the Physical Society for inviting me to host this virtual journal club. And obviously, Rosie Heinard, who helped us for the preparation of the meeting. And so please follow us and for the networking session that will start in a few seconds.
Thank you very much.
Thank you for having us.